Lecture since the series was reinstituted. It seems unbelievable that it could be that long, but it definitely is. Today's Buhl Lecture is Bill Gelbart, who served as Chair of Chemistry and Biochemistry at UCLA. And he's been talented enough to have two scientific careers. First of all, in the early 70s, Bill was a pioneer in the then emerging field of complex fluids, and he contributed significantly to the fundamental theory of liquid crystals, of self-assembling systems, polymer solutions, nanoparticles, and biological membranes. That would be enough to bring it here today, but it's not the subject of today's lecture because about a decade or so ago, he became fascinated by viruses, and he set out to understand their infectivity by investigating them outside their host. And that work is certainly truly multidisciplinary, involving both physics and chemistry and biology. And indeed, two of the postdocs that were in this lab, Alex Levinovich and Marcus Discerno, are today in the biological physics group in the Department of Physics at Carnegie Mellon. He tells me, Bill tells me also that his new recent research is the added attribute that when people sitting next to you on the airplane ask you what you're doing, they find it a lot more interesting to hear about viruses than complex fluids. His research has been recognized with many awards, including the 1991 Leonard Jones Medal of the British Royal Society and the 2001 Liquids Prize of the American Chemical Society, plus election to the American Academy of Arts and Sciences last year. As with almost all of the Buhl Lectures, he's not only doing great research, but he's a great teacher, having been recognized with the University Distinguished Teaching Award at UCLA. It gives me great pleasure to introduce the 2010 Buhl Lecture, Bill Gelbart, who will tell us about one of the aspects of his foray into the beautiful symmetrical world of viruses, namely making viruses from scratch. Yeah.
Thank you. Good afternoon. It's a pleasure to be here. I was last here at Carnegie Mellon about 10 years ago, and as Fred pointed out, my life happened to turn a corner then. I learned enough about viruses to decide that it was time I tried the first experiment in my professional life, and that I think seriously about biology for the first time. I, as a student, was enamored of my father, my twin brother were mathematicians. It was a beautiful world I learned about growing up, and I wanted to do something different, so I became a theoretical physical scientist. But indeed, I was enamored of equations and beautiful theories. I still am, but I didn't realize what I was missing. I was convinced that a physical theorist couldn't contribute anything significant to biology because biological systems were just too complicated. There were no general laws in biology. I'm quoting my ignorant former self. So I managed in high school not to take the obligatory course in biology, same thing in college. So people think I'm being modest when I say that when I started 10 years ago working on viruses, I knew no biology. I mean, I knew no biology, possibly less because of the great effort I had made not to learn any. But when I learned what a virus was and how simple it is as a physical object, my professional life changed in that I realized I was determined to do anything I had to do, including start a lab, in order to understand viruses. Real viruses, not models of viruses. So of course, as a theorist, I continue to make models of viruses. It's how I understand anything I'm thinking about in science. But the models, the theories, are explicitly intended to be tested by an experiment that we're designing at the same time. So in our research group now, we have 6-8 PhD and postdoctoral students, and generally no more than one of them is working on a theoretical problem. The others are in the lab working on a different virus. Why on earth would we work on several viruses when we start out knowing no biology and virology? We wanted to bring to virology a different point of view. As a physical theorist, again, it was natural to look for general behavior. So I wanted to know, I still wanted to know every year along the way and today, what do all viruses have in common? What is virus? What qualifies something to be called a virus? So I'm going to go through a lot of history. I think half of my talk is the history of virology. But I hope you'll agree that after we go through that history, you will have learned some things about viruses. It happens my wife is a historian of science, and early on I learned that studying history of science tells you a lot about scientific ideas. And it's all the more true in the case of virology, because you have to ask right away, why are viruses only something we know about in the last century? The concept of virus was not clarified before 100 years ago. So I want to talk about that phenomenon and how much you learn about viruses by going through the early history, the prehistory of viruses. Viruses from scratch. Yes, one of our big efforts is to make viruses from purified components, in that sense, to make them from scratch. But I also intended it with another meaning. I'm starting from scratch, telling you about viruses, assuming you know nothing about them, assuming you know as little as I knew when I started. We'll be asking lots of questions. I warned you we'll be asking what are viruses? What's a virus? What isn't a virus? And in particular, what about them accounts for why they were only discovered 100 years ago? A question that often comes up is, are viruses alive? We can talk about that after the lecture. You can ask questions about it. It's an interesting question, but one can also argue it's not important. Instead, talk about what they do. I argue it's useful not to think of them as being alive, and that helps you understand why they can be such simple, evolving organisms, if you like. They evolve no differently, in fact, much better than truly living things, faster and better. But they can have as few as 1 or 2 genes. The business of life is much more complicated than what can be coded for in 1 or 2 genes. You know that there are many proteins that come from the same gene. So there's a huge number of proteins that function in different ways to keep us alive, keep us healthy, compared to the number of genes that are identified in a genome. A virus can have 1 or 2 genes, so you have to ask, what are they for to allow the thing to act as a virus? And that gets us into the question of what are the generic life cycles? I'm going to show you 3 life cycles. There's a life cycle for every virus, and there's an essentially infinite number of viruses. We were talking earlier today about how easy it is to discover a new virus. Dig up some dirt, look under the microscope, and you'll see some new viruses. But why will I show you only 3 life cycles? Because maybe that's already too many. You see that they all have something in common. So we'll get to the heart of what a virus is by looking at life cycles associated with viruses that infect bacteria or plants or animals. That's as different as their hosts can be, so you'd expect the biggest differences in those viruses. And yet there are dramatic similarities, profound ones, between viruses that infect totally different kinds of hosts. This is one of the questions that drew me into viruses as a theorist. and it accounts for why viruses as physical objects are as beautiful as they are. I want to insist, I apologize beforehand for talking about many times throughout the lecture how beautiful viruses are and so on. We know that they're ugly, insidious, deadly, painful, horrible disease agents. So when I talk about viruses being beautiful, I'm talking about the virus particle. Okay? And related to these other questions, why, how can we make viruses from purified components in a test tube? You can't make a bacterium or any other one-celled or more complicated living organism from scratch. You don't know what the parts list is. You don't know what to begin with. And there's no way you could control all the conditions necessary for the evolution of that living thing. Simplest viruses are just protected genetic information. So the simplest virus, at the very least, has to have a genome that codes for at least one protein. Unlike all other evolving organisms, and certainly all living things, the genome needn't be DNA. We talk about DNA as the genetic material. Most viruses have as their genetic material RNA, single-stranded RNA. They need to be protected. And they're protected by a one-protein thick shell, which in most cases is spherical. In most of the cases, when the shell isn't spherical, it's cylindrical or rod-like. Sometimes you have a combination of the two, like in bacterial viruses, as I'll show you. We speak about a head and a tail. It's really a spherical head that protects the genetic material and a tail for delivering the material. And in the case of many mammalian viruses, certainly human viruses, there's an extra layer of protection. It's a lipid bilayer envelope that surrounds the protein shell. And that's important for the virus to get into and out of cells. Everybody knows what double-stranded DNA looks like. I was walking through the campus holding one of my favorite DNA duplex models to bring to a friend, and there were some 5-year-old kids playing, and one of them called out to their father, "Look, Dad, that guy has a DNA." So this is an icon, really, of biology, the double helix. What people are less familiar with is single-stranded DNA or single-stranded RNA. It's essentially the same polymer, but with some crucial differences. A ribonucleic acid, meaning a ribose sugar rather than a deoxy sugar, but that's the difference between a H and a OH group. And then one of the nucleotides, one of the bases, is different. The T of DNA of the 4-letter alphabet is slightly different. That small difference makes a difference. But the important point I want to emphasize from the outset is that the genomes of most viruses are single-stranded RNA. So it's a theme of our research to know the physical properties of each of the genomes. A lot is known about double-stranded DNA as a polyelectrolyte, as a physical object. It's a stiff polymer. It's been studied from every point of view. But single-stranded RNA is much less well understood and much more complicated. Let's start asking some physical questions. How big is a virus? When I teach, I like to insist that you can make an order of magnitude estimate of anything if you understand just a little bit about it. How do you figure out how big a virus particle is, whether it's a micron or an angstrom? Well, it clearly has to be small compared to its host cell. And a host cell is at least microns in dimension, so a virus has to be very small compared to that. Notice already we need to know something about biology. We need to know the size of cells. And we also need to know that when a virus goes through a life cycle and is replicated, you go from one virus to thousands or tens of thousands of virus particles. The size of a viral litter is on the order of 1,000. The gestation period is hours. That's one of the reasons they evolve so fast and out-evolve their host, okay? Or the immune system, or prescribed antiviral treatments, and so on. So it's got to be small compared to this, and yet it's got to be big enough to enclose a few genes. So let's ask how big a gene is. You saw the familiar double-stranded helix. The helical repeat, 10 base pairs, is 3.5 nanometers. The width of the diameter is a couple nanometers. So you can ask, if you need, say, 1,000 base pairs, and that's roughly what you need to code for a small protein, how much volume do you need to accommodate that amount of genetic information? And then ask, well, what about 10 genes? Because 10 genes is the typical genetic content of a virus. And it turns out you need a volume of tens of thousands of cubic nanometers. And you ask, how big a sphere would you need for that? A sphere with a diameter of tens of nanometers. So it tells you a virus should be tens of nanometers in size. And the remarkable fact is that all viruses are this size. There are big ones and small ones. And there are always exceptions. It's a few outliers. But to within a factor of 2, the overwhelming majority of viruses are 50 nanometers in size. That's why nobody ever saw them under an optical microscope, the way you see a bacterium. It's also why they couldn't be filtered out of a body fluid when the microbe hunters in the last century were looking for microbes, disease agents. They took blood, lymph, feces, and ran it through a filter, and the bacteria got caught in the filter. And they discovered lots of disease agents that way. They didn't discover viruses that way, because even the finest filter wasn't fine enough to catch a virus. So I mentioned that viruses were only discovered 100 years ago. The first one, tobacco mosaic virus, was discovered in the late 1890s. That particular virus plays a very important role in the history of virology. It was the first to be crystallized. That was in the 30s. And remember, people didn't know much about what a virus was then in terms of protein, nucleic acid content, how it worked. But they did think of it as alive. And no living thing had ever been crystallized. It's hard enough crystallizing proteins, those of you who do protein crystallography. Try crystallizing a bacterium or a microbe. Forget it. You need a highly monodispersed, same size, same shape, regular thing to crystallize it. Living things are not highly monodispersed. We have no symmetry. You know, we talk about people having right-left symmetry. We do approximately, but we have no symmetry in a strict sense. A protein molecule has no symmetry. But it turns out that a virus particle does, and it has the highest possible symmetry. It's an aggregate of proteins. So it's the first to be crystallized. It was the first to be reconstituted from purified components. That was in the mid-50s. I'll talk about that shortly. And again, nobody had ever heard about reconstituting a living organism. And I was suggesting we never will. First animal virus was discovered about the same time. Foot and mouth disease virus. The first cell cultures were developed for animal virus plaque assays in the early 50s, and that revolutionized virology and medicine. First vaccines in the mid-50s. First serious discussion of using viruses as vectors for delivering genes that were missing or defective in cells started in the 70s. First virus that infected bacteria was discovered in the course of World War I. I want to tell you that story in a moment. Viruses, and in particular bacterial viruses, called for short bacteriophage or phage, played a vital role in the origin of the field of molecular biology. Max Delbruck and a few others in the 30s and 40s realized that the virus was a simple enough system for unraveling on a molecular level the way in which genetic information is replicated and expressed, the way in which genes are expressed. That was done, the model system, so to speak, was a bacterial virus. And that was the beginning of molecular biology. The first EM pictures of viruses were in the very early 40s. I want to say a few more words about history. Maybe you're familiar with Kochs postulates, German biologist, medical scientist, who, again, certainly revolutionized bacteriology and medicine, discovering the disease agents for tuberculosis, many other diseases. And he got serious about how you hunt a microbe and establish that it's the causative agent for disease. You have to be able to find it in diseased tissue in every instance of the disease. You have to be able to isolate it, purify it, and grow it in cell culture, or rather sorry, I'm thinking about viruses, jumping ahead. You have to grow it in nutrient broth. Just give it some food and it'll grow. It's alive. Grow it, and then infect healthy tissue with it, and find the same disease and recover the same disease agent. So this made the 19th century, especially in Europe, the century of microbe hunting. It was a very exciting time. Pasteur got involved. But the reason viruses weren't discovered is for the reasons I already have hinted at. They were too small to see in a microscope once you isolate them. They're too small to be caught in a filter. And when you try to satisfy another one of Koch's postulates, grow it up in nutrient broth, it doesn't grow. That's because it needs to be in its host. It's an obligate parasite. It's not an independent organism. This is a picture of the kind of porcelain filter that was used. You forced with a hand pump body fluid or ground up leaves if you were looking for the causative agent of tobacco mosaic disease. Chamberlain was an assistant of Pasteur who developed this. Viruses were discovered only when people started looking in the filtrate, what got through the filter. It's interesting that cholera was discovered in the Koch sense, and inoculation was developed against it by doing just this. An interesting story, it was discovered by accident. Glad to talk to you about it later. What I want to stress now is that Pasteur was famous for many things, inoculating against cholera. Cholera is a bacterial disease. He's famous for inoculating against rabies. Now, rabies is a viral disease, but he didn't know it. And earlier, Jenner, in the late 18th century, had inoculated against smallpox. He didn't know that it was a virus. He didn't know it was different from a bacterium. This was long before Koch. Okay? This guy, who's not as famous as Pasteur, I think should be given credit for discovering the difference between a virus and a bacterium. He was a bacteriologist, so he tried to, he isolated the, he collected the filtrate from ground up leaves from plants infected with tobacco mosaic disease, and he couldn't grow it in nutrient broth. He found he could only grow it if he reinfected plants with it. And so he made that distinction. The word virus had been used by Pasteur and others. It's from the Latin poison. So it was a generic term. At the same time, these German medical scientists discovered hoof and mouth virus by looking at the bacterial-free filtrates of infectious material from sick cattle. And they point out that the causal agents of a large number of other infectious diseases, such as smallpox, cowpox, scarlet fever, measles, typhus, cattle plague, etc., might turn out to be similar organisms. Durrell and Twort in England discovered bacterial viruses, and I like this account by Durrell of his discovery. He was a medical doctor treating an outbreak of dysentery. It's a bacterial infection, an unpleasant intestinal infection. And he says, I made an emulsion of some of the still bloody stool and filtered it through one of these porcelain candles. And to a broth culture of dysentery bacillus, meaning the bacteria causing the dysentery, that he had isolated a few days earlier, I added a drop of the filtrate and spread a drop of this on agar. The next morning, on opening the incubator, I experienced one of those rare moments of intense emotion. This guy was serious, as we all are. I mean, which reward the research worker for his pains. At the first glance, I saw that the broth culture, which the night before had been very turbid, was perfectly clear. All the bacteria had vanished. Of course, they hadn't vanished. They had been infected by the virus, and I'll show you a picture of what a bacterial cell looks like infected by virus. It's filled with thousands of virus particles. And the only way they get out is by lysis, or breaking of the cell membrane induced by one of the viral gene products. Okay? So it's a part of the life cycle to destroy the cell membrane of the host so that all the new copies of the virus can get out. So when you lyse the cell, you can no longer see it under an optical microscope. This is the first EM picture. It's not very impressive. There it is. That's the bacterial virus. That's the bacterium. Here's a scale bar. These happen to be pictures that Alexey Vilevich took 65 years later. You can see a lot more of what's going on. Apparently, in the late 40s, Roger, correct me if I'm wrong, I think that's when Gelbart first saw an EM of a phage and exclaimed in German, "My God, they have tails." That certainly wasn't apparent here, but it is in a better quality picture. I put a scale bar in here, but I like to be mischievous, perverse. I show electron micrographs without putting in a scale bar. If it's a virus, you don't need a scale bar. They're all 15 nanometers, okay? Modulo a factor of 2. But to understand what's going on, that's good enough. Here's my favorite picture. Happens to be 3 tailed bacterial viruses tricked into thinking that this closed lipid bilayer that's been reconstituted with a few molecules of the receptor for this virus, it thinks, but it's not thinking. It's not alive. It's diffusing around like a Brownian particle. Its tail hits that receptor protein, and that triggers the release of the genome into the aqueous buffer solution inside this vesicle. We happen to put some polyvalent cations there to condense the DNA. And indeed, this is the kind of DNA structure that I was working on in the late 1990s before I knew what a virus was. I didn't even know what the person in the street knows about viruses. Another one of my idiosyncrasies is that I don't read the newspaper much or watch television at all. And so I miss a lot of good things, like the health section that tells you a little about viruses and has pictures. But this is 3 molecules of double-stranded DNA, 3 genomes from these viruses that have been condensed, hexagonally close packed by polyvalent cations. I'm exaggerating here. It might only be 15 orders of magnitude simpler than any living object. And it's because, I would argue, they're not alive. They have no metabolism. They don't grow. We speak about maturation of virus particles. But effectively, all that happens in a viral life cycle is nothing at all until they're replicated, and you go from 1 to thousands of copies in a proper host cell. Nevertheless, I've already emphasized that viruses have evolved to do precisely this in an alarmingly effective way. They get their genetic information into a host cell. They pirate the biochemical machinery of the host cell. It's the ultimate challenge for them and for us as scientists to understand regulation of gene expression. Viruses are all about regulating or misregulating gene expression. In an infected cell, essentially all the genes being expressed, and therefore all the proteins being synthesized, are those of the virus, not the host cell. That's why your host cells are sick and why you can die very quickly. Because within hours, you go from one virus in a cell to thousands of particles being made, each of which infects another cell within hours. I emphasize again. Let's go through the textbook pictures of viral life cycles. I'm sure everybody's seen pictures like this, including in the health section of the newspapers. Here you have, this is a schematic of the viral shell with the genome inside, the lipid bilayer envelope around it, and then transmembrane proteins that are important for getting the virus in and out of cells. It comes in through a process called endocytosis, where it picks up another layer of envelope. Might also come in by the two envelopes fusing. But the important point is that the naked capsid is delivered to the cytoplasm. The genome is released. A large number of copies of the genome are made, maybe millions. You then express the genes of that genome, and you make still more copies of structural proteins, like the proteins that make up the capsid. And then you make thousands of copies of the virus, and they get out, in this case, by exocytosis, by picking up their membrane, by stealing a piece of membrane from the host cell. In the case of a plant cell, essentially the same thing happens. All viral life cycles are the same in that they deliver the genome, large number of copies of the genome are made, and then you make still larger numbers of the structural proteins of the virus. The fundamental difference in a plant cell is that there's a rigid cell wall. So you don't have a delicate membrane protein or receptor-mediated entry process. The infection of the cell depends on slight injury to the cell wall. A leaf might blow against the trunk of the tree. An insect might be nibbling on one end of the leaf and do some damage to the cell walls, not enough to hurt the cell much, but enough for a large number of virus particles to come in and start an infection. In the case of bacterial viruses, again, it's the same fundamental cycle. Genome comes in, many copies made, genes expressed, many structural proteins made, many copies of the virus go on to infect other cells. But this is the first life cycle we looked at. And as I was reading undergraduate textbooks trying to learn about biology and about viruses, I saw this cartoon and I asked the simple question, what drives this ejection or injection of the genome? The cartoon showed the virus particles staying outside the cell. In the case of the plant and animal viral life cycles, the whole viral particle comes in. And that means you have to find a way to understand what drives the delivery process. This is a famous experiment that was done in 1952. It's cited most often as providing confirmation that DNA is the transforming material, the genetic material. But in fact, for our purposes, what's important about it is that it proved that the virus particle, the capsid, stays outside. The protective shell stays outside. You radio-label the protein, you radio-label the DNA, and midway through the infection process, you put your sample in a kitchen blender and knock off the capsids and pellet the cells, collect all the cells in a centrifuge, and what you find is that the only radioactively labeled material is DNA. Only the DNA has come in, no protein. So again, it points up the fact that something drives the DNA in. And I just show that same picture here, and the question we ask. Here's an electron micrograph of a fairly infected cell. There are barely even 100 particles here. There's room, and you're just seeing in the plane of focus. Before this cell has its outer membrane broken, lysed, there will be thousands of particles made, each one going on to infect another single cell. In this case, it appears there were two viruses that injected their DNA to start the infection. It's been recognized by several people. This is a cartoon we had drawn for a commentary last year, making an analogy between how a bacterial virus delivers its genome and how a mammalian or human virus delivers its genome. I had said before that, and emphasized just now, that it's, with a few exceptions, only the bacterial virus that stays outside the cell. In the case of plant and animal viral infections, the whole virus particle comes inside. So you don't have the problem of explaining how the genome gets inside. In the case of herpes, which you know all too well, it is a very successful predator of humans. It's an enveloped virus, which means you have the genome, you have the protein shell protecting it, and then you have the lipid bilayer, a piece of membrane, fluid membrane protecting it. You have fusion of this membrane with the cell membrane. You have a naked capsid that is trafficked through the cytoplasm to the nucleus. The genome is DNA. A DNA virus has to get its DNA into the nucleus. That's where DNA is replicated, and that's where genes are expressed. And yet, the analogy between the two is that the capsid doesn't enter the nucleus. There's a membrane around the nucleus. There's a pore complex, and binding to the pore complex triggers the injection of the DNA. So there's reason to believe that it's the same physical mechanism that drives these genome deliveries. And that mechanism is pressure. This is a famous photograph from many decades ago. It's an electron micrograph of a bacterial virus that's been broken, in this case by osmotic shock. What you see here is the genome that had been protected inside the head. Notice I have no scale bar. The head is 50 nanometers in diameter. This DNA is tens of microns long, so it's many hundreds of times longer. This dimension is a micron, so it's tens of times bigger than the head. And this is a single molecule of DNA. You're used to looking at DNA as a double helix on an atomistic level, but this is the double helix. And it's a pretty stiff molecule because of the base pairing and stacking. And in fact, you know what 50 nanometers looks like. You'll never see DNA spontaneously bending into a radius of curvature smaller than 50 nanometers. That's the persistence length. That's the 1D elastic modulus of double-stranded DNA. And this is one of the ways to measure it. You just see, by definition, only thermal energy is available to bend this DNA, and that's why it's never bent into too small a radius of curvature. And yet, all this DNA, which has 2 charges per base pair, 2 phosphates on the side chains per base pair, so it's got a very high charge density along its length. It's hard to bend, and yet it was confined at close packing densities inside this shell. So we asked the simple question, how much work do you have to do to take double-stranded DNA and confine it this strongly? And you can ask how much work you have to do. You can ask what the stored energy density is. That's effectively the pressure. And we found a surprising result. It was 50 atmospheres. And it was hard to believe, so we knew it had to be measured. And the first experiment we did in our lab was done by Alexey Vilevich. We didn't have a lab when he came, to his credit. He built the lab and did this experiment. And everything depends on this fact about the relative size of the viral genome and the viral capsid. Now remember that the capsid, I haven't said it yet, is formed by the self-assembly of one of the gene products of the virus, namely the capsid protein self-assembles to make an empty shell 50 nanometers in diameter. Another bunch of viral gene products makes certain that you get replication of the genome to a high level. Some other gene products make certain, including a very strong protein motor, that you can do enough work to stuff this DNA into the capsid. Okay? So why would it be that a virus evolved to depend on so much work being done, building up such a high pressure? Things that evolve are things that survive. The only viruses that survive are ones that are strongly enough pressurized to deliver their genome to a host cell. This picture, so Roger, this is the picture I wanted your reaction to. This is a picture we took just this year, a postdoc in the group at J. Gopal. This is an electron micrograph, a cryo-electron micrograph, of the viral RNA genome of particular plant virus we work on, cowpea chlorotic mottle virus. And this is the analog of the picture you see here. We are showing, juxtaposed next to each other, the viral genome and the capsid inside which the genome is confined. You notice that this RNA molecule has the same dimensions as the volume in which it's confined, consistent with the fact that this is just a solution of purified RNA we threw in some virus. But what we normally do is to have purified solution of RNA, no virus around, and we throw in some purified capsid protein. The capsid protein that is one of the 4 gene products of this simple virus. You mix those 2 purified components in pH 7 buffer, and they spontaneously organize themselves into infectious virus particles, not just spherical, but icosahedrally symmetric shells, 1 protein thick, 180 copies of the protein repeated to make the shell, and 1 and only 1 molecule of the RNA inside. So this genome gets packaged, protected by a spontaneous self-assembly process, whereas the double-stranded DNA virus has its genome packaged and protected by a lot of work being done by arguably the strongest motor protein known, the gene product of that particular virus, the bacteriophage or herpes, whatever. So that's a fundamental physical difference between the genomes of double-stranded DNA viruses and single-stranded RNA viruses. And that's how we try to understand how viruses work and why we call what we're doing physical virology, because instead of asking genetic or biochemical questions about the virus, we're asking physical questions in order to understand how the virus works, what makes it infective. So again, it's this fact which had been suspected but never demonstrated directly. We had done several years of work with standard methods for determining the sizes and shapes of large polymers, biomolecules like viral genomes, small angle X-ray scattering, diffusion coefficient measurements, but it didn't give us direct information about size and shape that we get in this electron microscopy. So I've just summed up what I've told you so far, how we think about the physical difference between DNA and RNA viruses. Basically, we say it's no surprise that viruses that don't enter the host cell but rather deliver the genome need to be pressurized. Therefore, they need a double-stranded DNA genome. Double-stranded RNA would work too, but the large majority of viruses, bacteriophages, that depend on pressure, have double-stranded DNA virus genomes. The large majority of plant and animal viruses have single-stranded RNA genomes because they don't depend on the genome being stiff, big, and hard to confine without a lot of work being done. In fact, they depend on the genome being spontaneously packaged through self-assembly with the capsid protein. We've been working the last few years on enveloped viruses, and in particular trying to make one from scratch. It's something I'm glad to talk about afterwards. We have some results after 5 years, but we haven't yet succeeded in making from purified components. Let's see, it's 1, 2, 3, 4, 5 components. RNA genome, capsid protein, 2 membrane proteins, and lipid. We haven't succeeded in making the full infectious virus yet. Only halves of it. We can't quite put them together and get the infectious virus. I mentioned tobacco mosaic virus before. It's the first one discovered, the first one crystallized. This is what it looks like. It's a remarkably same size, same shape virus. Happens to be rigid cylindrical rods, hollow, 1 protein thick shells with 1 molecule of RNA inside. And this was reconstituted in infectious form from purified capsid protein and RNA. That was done in 1955. A decade later, a similar experiment was done with purified capsid protein and RNA genome from a spherical virus, a simple plant virus. In fact, the one that we do most of our work on. Establishing that this is a spontaneous self-assembly process and that, yes, you depend on evolution to give you a protein that will spontaneously self-assemble with its genome, but it needn't take place in the host cell. You don't need host cell cofactors. I want to come back to the kind of physical questions we ask about viruses to understand them. Once we know how big a virus is, we settled that question earlier by doing a simple calculation. Now we want to know typically how many capsid proteins do you need to make up the shell. And you know the size of a protein. It's a few nanometers. You know a typical radius for the shell. Therefore, you can find that there are on the order of hundreds of proteins you need to make a 1 protein thick shell that's 50 nanometers in diameter. Okay? How much RNA can fit inside? Well, you can figure out the volume inside. Suppose you had RNA at maximum density, 1 gram per cc, 1,000 Daltons per cubic nanometer. It would tell you how much RNA you can have inside. That corresponds, it turns out, to just a few RNA genes. So this is the way we figure out how many genes a virus has typically. It has on the order of a few. Big viruses have hundreds. Small ones have a few. Typically, you have tens of genes to do the job of this generic life cycle I showed you. There's a remarkable paper by Crick and Watson, a 3-page paper in Nature, 1956, nowhere near as famous as their 1953 paper, Watson and Crick, on the structure of the double helix. but I think it's a more impressive paper, period. I tell my theory students it's one of the more impressive pieces of theory done in science, period. Because without any equations, without any computations, with some deep thinking about biology, and this was before the genetic code was deciphered, before people knew about messenger RNA, the central dogma, how you go from DNA genes to protein products and so on. They realized that there's only room for a few genes. They had an idea of how big a gene had to be, and they realized immediately the capsid must be made up of many copies of a single gene product, instead of having many genes coding for many proteins of different kinds that would make up the shell. There isn't room to code that much information. So waste only one gene on the capsid protein and try to understand how a symmetric shell can be formed from a single molecule. And then they did some basic group theory without calling it such, and they figured out that the symmetry of viruses should correspond to cubic point groups. Basically, their idea was that if a single kind of molecule makes up the shell, then for the shell to be stable, there should be some optimum way for that molecule to be surrounded by neighbors. All the molecules should be similarly surrounded by neighbors to have a similarly low energy. They didn't even talk about low energy. I'm putting a lot into their argument. That's the genius of their paper. So you need all the proteins to be in equivalent positions so they see the same neighbors, nearest neighbors, next nearest neighbors, and so on. And if you ask yourself, how many points can you put on the surface of a sphere so that the positions are equivalent? Not sort of the same, like the dimples on a golf ball. Take a look, they're not equivalent. You can't put that many points on the surface of a sphere and have them strictly equivalent in the mathematician sense. So this begins to sound like a geometry problem, and it is. The answer to the question is that the maximum number is 60. Sounds counterintuitive. You'd think if you just spread them out uniformly and carefully, you could go on forever. But it's also counterintuitive that there's a limit to the number of perfect polyhedra. There are only 5, and the icosahedron is the biggest one. Those are the platonic solids. You would think if you take regular polygons and you're careful, you can make an arbitrarily large polyhedron where all the faces are the same and each is a regular polygon. But you can prove that there are only 5 and the biggest one is an icosahedron. There are 20 equilateral triangle faces. Symmetrically put 3 points on each face, you'll have 60 equivalent points, all the same distance from the center and all equivalent to each other. That's the prediction that Crick and Watson made. And just reminding you that an icosahedron is the highest point group symmetry. You have lots of rotation axes. You can't have a center of inversion. You can't have mirror planes because this is made up of proteins. Proteins have no symmetry. You have chiral atoms. Think about it. Proteins have no symmetry. But you can have aggregates of them that have this high symmetry. A dodecahedron, 12 pentagonal, regular pentagon faces, again, it has the same symmetry as the icosahedron. The mathematician says these are duals of each other, same symmetry operations. And again, put 5 points on each face symmetrically, and you have 60 equivalent points. And that's what viral capsids look like. Each face has proteins. You have nothing along the edges and vertices. The faces are made up by groupings of identical copies of a single gene product. What Casper and Klug did, again, 2 great structural biologists, in a single paper, they didn't write a series of papers, there was a single paper in which they solved the rest of the problem. Okay, if you can have 60 proteins that are equivalent, you'd say, okay, protein shells, to be most stable, will have 60 proteins. But that just gives you a capsid of a certain size. Remember, all proteins are a few nanometers in size. You want to make something that has twice the diameter, you need a lot more proteins, okay? They won't be equivalent any longer. So these guys asked another geometry question. The shell is essentially a 2D crystal. Proteins have aggregated in 2D, but it curves on itself. It's a hexagonal crystal. It has that symmetry. And so it's a geometrical problem. Descartes and Euler solved it centuries ago. How do you take a hexagonal crystal and close it on itself? You need to introduce 12 five-fold defects or disclinations. Or if you like, you need to take hexagons. Let's see. I made this picture up before I left home the other night. This is my newest favorite toy for model building, Polydrone. You try to tile the floor with hexagons, no problem. You try to do it with pentagons, you can't. Okay? There's an angle deficit there, and it can only be relieved if you want a closed shell by coming out of the plane. And so all the curvature comes from pentameric groupings of proteins. And in this way, Kasper and Klug figured out what the sizes of the bigger viruses would be. There would be very discrete sizes because you either take one step along this direction and one step along that other lattice direction and put another pentameric grouping, or you take more steps along one direction before you do it. You repeat the process labeled by these two integers, how many steps you take along each direction, 12 times, and you've made a closed shell. I quickly went through them. Now let's go a little more slowly. And I'm almost finished. Here, indeed, the simplest, smallest spherical virus has 60 copies. It is, if you like, an icosahedron, or equivalently a dodiagonal pentahedron. Bigger viruses, like our friend herpes, you have to take 4 steps along one direction and then none along the other before you put in a pentameric grouping. Remember thank you, Alex. Can everyone see this? Everybody knows what a soccer ball looks like, an old-fashioned soccer ball with 12 black pentagonal faces and 20, most soccer players aren't aware of this. If you see people playing ball, be sure to stop them and make a nuisance of yourself. 20 white hexagonal faces. This is the next biggest virus particle that's seen. You take one step along one lattice direction, one step along the other, and you get this structure. And each white hexagon is a hexamer, a grouping of 6 copies of the capsid protein. Each pentagonal face is a grouping of 5 copies of the same protein. And when you do the math, you see you have 180 copies of the protein. The next biggest will have 240 copies. The next biggest will have 720 copies. And that corresponds to a discretized set of diameters. In the case of herpes, by the way, this T number, that's the sum of the squares and the product of the number of steps you take along each lattice direction, it's telling you how many inequivalent positions you have for these identical proteins. So in the case of the soccer ball, when you fill up the faces with proteins, you find that there are only 3 inequivalent positions. You want to minimize that number. That's the problem that Kasper and Klug, as structural biologists, solved. It's a geometry problem, but no geometer had thought about it because he or she wasn't thinking about viruses in a deep way. My last few slides now. Maybe you've seen these pictures. Quickly, I go to some more here, but I'll come back. This is, again, herpes. These are ugly diseases, but this is adenovirus. This is a bacteriophage lambda, that we did our experiments on, that played a big role in the beginning of molecular biology, in the genetic engineering revolution as a vector for cloning. But you admit every one of these particles is beautiful. They're beautiful because they have the highest possible symmetry. No living thing has that symmetry. Okay? This is the plant virus we work on, which has 180 copies of the protein, like the soccer ball structure. This is papilloma. Here's polio. Again, ugly diseases, beautiful particles. And you remember, R is the diameter or radius. The genome, when it's inside, is essentially close packed. And you can easily show that, as a consequence, the radius scales with the cube root of the genome length, or the number of genes. The T number, the number of inequivalent positions, which is also telling you how many capsid proteins you have, the number 60 from platonic solids. This is not numerology. This is I insist, deep science and geometry. 60 times the number of inequivalent positions is the number of copies of the capsid protein, and the size scales as the square root because the area goes as the number of proteins. So I think it's an appropriate place to end because it points up how treating viruses as physical objects tells you a lot of very germane and vital things about viral life cycles and therefore what makes viruses infectious. I've collected some of the conclusions on this slide, and I don't care so much about names, and we're talking all day long and many interesting discussions with the biological physics group and non-biological physicists, but the importance of doing interdisciplinary science, and that means you shouldn't be too quick to put labels on what you're doing. Don't insist that this work should interest a particular community. Rather, insist that you're asking an important question and you're getting a good answer to it. Then you're doing good science, whether it's interesting to mainstream physicists or chemists or biologists, whatever. I think it's pretty clear that the study of viruses, like the study of material science and so many other problems in science in the 21st century, has to be approached from a broad interdisciplinary point of view. I generally count on questions during a talk. I realize it doesn't work in this kind of venue, but I understand you have questions afterwards and that there's a reception where we can talk more about any questions you have. Thank you very much. I'm sorry, what does? Yes, oh, sure. Yeah. It does and it doesn't, yes. I think, so the question was, what's the connection between, I mean, most people throughout the world know this as a soccer ball. All the nerds in the world know it as a buckyball, right? This is carbon-60, Buckminster fullerene, and you can't see, but you can imagine the stitching. It's a nice old-fashioned ball. It's not a molded plastic one. So you have edges and you have vertices. Do you know how many vertices there are? There are 60. That's why this is that's why in the 1985 paper announcing the first fullerene, C60, there are 3 pictures: a high-resolution spectrum, the apparatus, molecular beam apparatus, and a soccer ball. So even then, the carbon atoms are at the vertices. I told you that the proteins that make up the capsid protecting the viral genome are not at the vertices or edges. They make up the faces. They make up the planar faces, 5s and 6s of them. So I think the chemistry and physics of how the fullerenes are formed is fundamentally different from the chemistry and physics that drives the self-assembly of capsid proteins to form a structure with the same symmetry. We got this symmetry, again, before we started doing experiments. We're just asking theory questions. We asked, what's special about icosahedral symmetry? We took the idea of Crick and Watson and Casper and Klug, and they had already figured out the hard thing. So we said, let's take their idea literally. Let's put points on the surface of a sphere and have them simply attract each other isotropically. They're confined to the surface of a sphere. If they get too close, they repel each other. That's the generic interaction between globular proteins. Okay? What's the lowest energy configuration? Well, keep putting more and more particles on the surface of the sphere, minimize the energy, and ask what's the energy per particle, and you find that it has an especially low value for certain multiples of 60, and it corresponds to icosahedral symmetry. That's the physics behind a capsid looking like a soccer ball. It's electronic structure of carbon that's behind the geometry of C60. And nothing against the fullerenes, but they're ugly compared to viruses. C60 is exceptional as a highly symmetric fullerene. You have a lot of lower symmetry fullerenes. You don't have many lower symmetry virus particles. Of course, it's biology, so you have exceptions to any rule, and there are some ugly-looking viral particles.
What drives the difference between these spherical or others?
Yes, good question. Why? I remarked that most viral capsids are spherical and icosahedral. Of the ones that aren't, most are hollow cylinders. So it turns out that those are the two ways to have many copies of a protein in the minimum number of inequivalent positions. In fact, in a helix, all of the molecules can be in perfectly equivalent positions. Crick and Watson pointed that out, and that's independent of how long it is. So we could have lived in a world, things could have evolved so that there were only spherical viruses. But it turns out you can, with a single curvature throughout the full tube, no matter how long it is. By the way, if you take the viral genome of tobacco mosaic virus and you cut it in half, as long as you leave a certain small sequence intact, and then you add the capsid protein, you will get exactly the same hollow shells, same diameter, but half as long. So the RNA is a template for how long the rod is, but the curvature, the fact that you form a hollow rod rather than a sphere, has to do with the subtle aggregation properties of the protein. Viral capsid proteins generically aggregate as a two-dimensional crystal. And the curvature they prefer in closing to protect the genome varies from virus to virus. So the plant virus I showed you, nice spherical capsids with 180 copies, we routinely make it in the lab. We can replace the viral RNA by a bigger RNA molecule and get a bigger capsid. So this is a capsid protein. The assembly process isn't as efficient, but this protein prefers a certain curvature, and evolution has made certain that the viral genome is the right size for the curvature preferred by the laterally assembling gene product, its capsid protein, but it can also form, with less efficiency, a larger capsid or a smaller one. In the case of tobacco mosaic virus protein, you can put in any RNA you like, it'll always form the same tube. There, the preference for that curvature is overwhelmingly strong. But basically, it's a quirk of evolution. that may be redundant because evolution proceeds through quirks, random mutations. One of these two geometries works for one virus and the other works for most others.
Thank you.
Thank you.
One more and then
Is the high internal pressure of capsid unique to viable material, or are there other natural phenomena that have that?
It's a good question. I'm not sure I know the answer. It's a natural question, and material scientists have asked that question. Is there anything special about, again, the capsid protein evolved to prefer a certain curvature. You know that if you had a mutation in the gene for the capsid protein of one of these bacterial viruses, or herpes, so that it preferred to make a bigger capsid, you'd still have the same genome length, and when the genome was packaged, you wouldn't have a high enough pressure to deliver the genome. You would lose infectivity, and that virus wouldn't survive. Alex has done experiments where you ask, how much pressure do you need in order for the virus to be infective? You can change the pressure by changing the length of the genome, but keeping all the genes intact. You can also change the pressure inside by adding more salt to the buffer solution, and the solution permeates the capsid and changes the strength of the repulsions of DNA for itself, so you change the pressure that way. So the pressures probably can be as high as 100 atmospheres without the capsids breaking. However, even at the working pressure of tens of atmospheres, you always have failure of virus capsids. That's why if you store one of these samples for a few days, you find less infectivity. The capsids are failing. They're pressurized capsules that are failing, just the way high-pressure metal cylinders in your lab will fail after thousands of years, sometimes sooner than that. You have a shell under stress, so it'll fail. I don't know of other single protein thick structures in biology that have to withstand pressures this great. It's an interesting question.